Hello, everyone. Good morning, good afternoon, good evening. Welcome to yet another installment of the Global IMMUNOTALKs in 2022. And it's a real pleasure on behalf of all the organizers, well, to welcome you all, but in particular our speaker today, Thomas Korn from the Technical University in Munich. Now, it's a great pleasure to have you here, Thomas, and I'm very excited about your presentation to come, and I'd like to say a few words about you. Thomas is actually a neurologist, and he did his clinical training both in Germany, in Würzburg, but also in London. He then did his residency, became a proper neurologist, did his PhD then in 2000, or his MD thesis in 2000. Then did a clinical postdoctoral fellowship again in Germany with and decided then after that he needed to learn more and was more interested in preclinical work, and decided to do his postdoc with Vijay Kushru at Harvard, and that was a very, very spectacularly successful time. Thomas is one of the people who defined a better understanding of IL-17 producing helper T cells. Thomas then, after his postdoc, went back to Germany, became a physician in neurology at in Munich, and then received a grant, a big award that allows him to start his own lab, which then quickly climbed the ranks, became associate professor, and since 2015 he is full professor, and since 2019 he is director of his Institute for Experimental Neuroimmunology at the Technical University of Munich. Thomas received numerous awards. He holds an ERC Consolidator grant. He received the PETA Prize. There's so many things, and his contribution to neuroimmunology are numerous and super, super exciting what Thomas has done. Now, Thomas, before I let you start sharing your screen and give us your presentation, I have two more things to mention here. At the end of the seminar, what will happen is I will share once again the screen on how to get in touch with Thomas, because it's likely that there will be too many people participating today that we can't all ask questions. But you need to ask Thomas questions. You should ask Thomas questions. You're encouraged to do so. I will share in the end the best way of doing this. They will also be recorded, and you'll be on YouTube later on, so people can look at your seminar and engage with you via Twitter. So what we do here at Global IMMUNOTALKs, we ask each of our famous speakers a couple of loaded questions, and the one that we selected for you was, what was the most important decision in your career, Thomas?
Well, okay, so that's a tough one. Actually, you know, there's probably a series of decisions that you have to make, but for me, it definitely was the decision to do a postdoc in the US, because I mean, I was engaged in clinical rotation at the time, and I was actually about to drop science, honestly, because I, was overwhelmed with clinical workload. But at the same time, I felt that that was a true interest of mine to better understand cellular immunology in the context of autoimmunity. And I mean, okay, I had been offered a good position in Germany at the time, but I declined that one and, decided to go abroad to do a postdoc, which was, certainly connected with a lot of uncertainty at the time. But I mean, I don't regret it at all. It was really probably also the best decision that I made.
So let me just ask quickly, do you think it was important that you did a postdoc in general, or did it have to be the United States, because you couldn't have done a postdoc elsewhere?
It was definitely to do a postdoc in general. It could have been any place. I mean, I took a long time actually I took that decision very seriously. So I interviewed in a lot of labs, and then picked the one that I thought would, best fit my needs.
Cool. Yeah, you did well. Very good. All right. So then let's get started. Why don't you share your screen? We'll look forward to hearing what you want to show us.
All right. Thank you for the invitation. So I'm going to share my screen. Okay, so you can see it, right? Okay.
Yes, we can.
Okay, great. So thank you again for the invitation. It's a great opportunity to be part of this enormous lecture series. It's amazing, actually, the lineup of speakers, and I'm honored to be invited to this. So what I wanted to discuss with you today is actually a concept that has been emerging, I would say, in the last couple of years, which is probably a bit more advanced for regulatory T cells, but is also now emerging for effector T cells, and that is tissue imprinting of autoreactive T cells in our case, because we are very much interested in autoimmunity, and in particular in autoimmunity in the central nervous system. And in order to start this, what I wanted to share with you today, I basically have two different starting points. I have a more clinical starting point, which is, very dear to me in a way, because I'm also a clinical neurologist. And I have a more conceptual starting point, which I'll talk about in a few seconds. So this is the clinical starting point. So the disease paradigm that we are talking about is multiple sclerosis. And you know that multiple sclerosis has many phases, and it's very diverse. It's probably more a syndrome than a distinct disease. But what you can do in terms of just phenomenologically looking at this disease is that you have different clinical courses. Yeah, so you have a relapsing remitting course. This is probably what most people know. But then you also have something which is called a secondary progressive course, or let's call it progressive course, because there's also people that start with this kind of disease course, more progressive. So they don't have any relapses at all. And that's maybe about 15% of the patients. And also the ones that start with a relapsing course, some of them, probably less than previously thought, are going to become secondary progressive along when they go along their disease, and that usually happens after, let's say, 10 to 15 years. So this is what we can see, what we can observe. And so we have this relapsing phase and we have this progressive phase. And I don't want to make too much of a point now in terms of distinction between secondary progressive and primary progressive. It's just, progressive, no clinical relapses anymore. And then there was actually a cohort which was started in the beginning of the 80s in London, Queen Square. And now, so we are looking at 30 years now, more than 30 years of this cohort. And that's a very valuable cohort. And these were patients that were diagnosed with multiple sclerosis at the time. So they started their disease in the mid 80s and they were followed up now for 30 years. And what you can see here after such a long time is that about, maybe 1/3 of these people are still relapsing and are still doing pretty well. So they are, in terms of their handicap, so they are kind of doing okay. And then there is maybe 2/3 of these people that after such a long time are pretty bad off, because they accumulated a lot of neurological problems over time, and many of them are actually secondary progressive. And the best discriminator between these two groups of patients, and I'm not discussing all the details now, I just want to make the point that there is probably one good discriminator, and that is actually the amount of cortical lesions that these people have and already had early on. So this feature discriminates pretty well whether you are going to be staying okay or whether you are prone to be doing pretty badly. And so that already gives you an idea that we really need to understand and come up early on with better ways to stratify these patients. So that is certainly a big clinical need in terms of not so much treating, that is of course also important, but also discriminating and prognosing, making a good prognosis how people are going to do during their disease. And since we have MRI, this is very well reflected actually in the MRI that you can do early on during the disease. So this is, for example, a patient that has a relapsing disease course, and this one has a lot of white matter lesions, as you can see here, for example, the corpus callosum, and here, which is typical, which is thought to be typical for multiple sclerosis. Yeah, so these white matter lesions. But then we also have patients that have more cortical lesions, and with the improvement of imaging technologies, we can actually visualize this early in the disease. So this one, for example, here is a lesion that is close to or extends into the gray matter, the cortex. And it's kind of likely that this patient is probably going to be doing worse than this one. And this is what we are very much interested in, because that is very important for the patient to know, and also then, of course, down the line to select the right treatment for this person. And what the key of our interest is whether there is immunologic determinants of this, whether there is properties of the immune system that would predict or dictate, if you want, whether a person has lesion types like this or lesion types like this on the right side. So that's basic question. Is there immune system intrinsic determinants of this? And of course, there's a lot of concepts out there. And since we are very much interested in T-cell immunology, and there's actually good reason for this, because there's a lot of evidence to suggest that T-cells are the main drivers of the disease, of the lesions, of the development of the lesions, not so much executing the damage, but inducing or organizing the type of immunopathology that happens. And as you know, there's loads of papers to suggest that some specific T helper cell subsets might be involved in this lesion development. And it's actually a good hypothesis to have that these subsets of T helper cells actually induce different types of immunopathology that may actually have to do maybe with, organization of these follicle-like structures in the meninges, or that may have to do with the development of tissue-resident memory cells that reside in the tissues and may contribute quite a bit to the not only site, but also the type of lesion that we develop in the central nervous system. And I think if we get a better understanding of this, we would actually be much better in giving good advice to our patients, and also to have a better prognosis how they will be doing in the future. So this is just a quick example of how tissue-resident memory cells, and I'm only touching this briefly, could have an impact on the development of immunopathology in the central nervous system. So this is a study of Doron Magler in Switzerland, in Geneva, where we had the chance to contribute a little bit. And that shows that if you have in a model system, if you create tissue-resident memory cells by an early infection of these mice, in this case with LCMV, which is totally cleared from the CNS again, but which leaves the mouse with tissue-resident memory cells in the central nervous system, and then later challenge these mice with autoreactive T cells, then they will actually come down with autoimmunity only if those tissue-resident memory T cells are present. And they are present if this prior infection actually happened in early life. So early on in the life of these mice, during the first week, basically, of their life, if they get this infection, only then they generate sufficient amounts of tissue-resident memory cells that reside long-term in the tissue, which makes them then susceptible, which basically lowers the threshold of their susceptibility to autoimmunity. That doesn't happen if they get infected with LCMV in later life, so as young adults, for example, and then get the same challenge. That doesn't then lead, at least not to the same extent, to the development of autoimmunity. So this is interesting because it's a threshold thing, that determines the threshold for autoimmunity maybe, but also the site where these lesions are. And they are most likely here close to the ventricles, for example, because this is the site where the tissue-resident memory T cells reside. And the mechanism that was suggested in that paper is that those TRMs would actually produce CCL5, and by this contribute to the attraction of autoreactive T cells later on, and then create a fertile field for the development of immunopathology. So that is conceptually one of the ideas that I think we should really understand better in order to, stratify our patients better. So this is the clinical starting point. And the second starting point of the story that I wanted to share with you is actually more an immunologic starting point, if you want. And that has to do with the diversity of T helper cell responses. And there is a big discussion in the field. I mean, previously we had Th1 and Th2 cells, and then we had Th17 cells, and now we have a lot more of these T helper cells. And the question, of course, is, is this real? Or is this something we just, force in a way in vitro studies, or is this also happening in real life? And I would say, yes, it is probably real to some extent, at least, because it's very clear that you need cells with features of, for example, Th1 cells in order to have a very good intracellular host defense. So if you have intracellular pathogens like Listeria, where you need interferon gamma in order to harness macrophages to get rid of these Listeria, you need T cells that can actually produce interferon gamma. And the same is probably true for antigens that cannot be ingested. There you need a Th2 response in order to have good B cell help in outside B cell follicles, in order to then make these B cells become plasma cells to make antibodies to opsonize these pathogens that cannot be ingested. So you need Th2 responses, and you probably need Th17 responses in epithelial barriers. But you can see already that this is always associated with a very distinct function of these T cells, of these T cell subsets. And the question is, do we find this also in the context of a real infection or in the context of autoimmunity? And that there is two big concepts basically to explain that we almost never find pure Th1 cells or pure Th2 cells or pure Th17 cells. And one of these concepts is actually plasticity. So that means you have a pure Th cell in the beginning, and then that becomes something else, or is plastic and takes on features of other T helper cell subsets because of some environmental impact, for example, some factors in the environment that would foster this kind of plasticity. So that's one concept. And the other concept is that the priming of these T cells, these T helper cells in particular, is mixed from the start. So you have a mixed priming, and you have a continuum from more Th1-like and more Th something-like along a continuum. And this is something that has been really coming up with all the single cell data that are now available. And this is just one example from Diane Mathis and Christophe Benoit's lab, where they infected mice with these bugs here. And you can see that the clustering actually goes along the bug and not so much along the T helper cell subset. So it's clear that Citrobacter specific T cells are closer to each other, no matter whether they make IL-17 or interferon gamma, than IL-17 producers that are reactive against another bug like Salmonella. And you can see this here in this dendrogram. So that means that it is probably the environment that determines a mixed priming of these T helper cells. And that makes it, very complicated conceptually. But I think this is an important issue to deal with, and something we really should maybe consider and rethink our concepts also in our particular case of our paradigm. And I think that our paradigm of multiple sclerosis is actually perfect to look at this, because there is very good evidence to suggest that the T helper cells that eventually go to the brain and induce this immunopathology are primed in the periphery. So multiple sclerosis is not a disease of the brain, it's a disease of the immune system. And the priming, at least in the initial stages of the disease, happens in the periphery and probably is triggered by antigens that either cross-react or are mimics of autoantigens. So autoantigen-specific T cells are activated by mimics of this autoantigen. in peripheral tissues, for example, in draining lymph nodes of the skin or in draining lymph nodes of the gut, and are then aberrantly homing, if you want, to the central nervous system, where they then are reactivated by the real autoantigen, by antigen-presenting cells, and then induce the immunopathology. So the question is, can we define, based on the site of their priming, specific properties of these T cells then later in the central nervous system? And I think this is really something we should think about in order to better understand and stratify the different types of immunopathology in multiple sclerosis. So we try to do this, and one way to do it, it's not maybe not the only, but one way to do it is to label cells in vivo in different sites, to have a site-specific labeling of this. And then you can actually look in the brain and the target tissue. And here, in this case, you would not know where this cell eventually was primed or where it eventually came from as compared to the other one. But you would know it in this scenario, where you have labeled one site, in this case, a skin draining lymph node, that the red cell came from the skin. And then you can ask all kinds of questions. You can ask, does it home to different sites in the central nervous system, which is a big issue in neurology, because it makes a big difference if you have a lesion in the brain or in the spinal cord. In one way, one scenario you wouldn't even notice, maybe. In the other scenario, you would basically be unable to walk anymore. So this is a big issue. And also ask questions, what are specific effector functions of these cells based on the site of their priming? So this was the starting point of the story. And so we used, in this case, photoconvertible proteins that we could convert by light irradiation, so site-specifically, from, in this case, green to red. So this is a dendra protein that we specifically expressed in T cells. And then we can actually irradiate specific lymph node sites. Yeah? So for example, skin draining lymph nodes here, or gut draining mesenteric lymph nodes here. So this one can be accomplished by just irradiation through the skin. And here we have to surgically expose the mesenteric lymph nodes. But when we do this, we can basically, we have a protocol where we can photoconvert basically all the T cells that reside in that mesenteric lymph node, or all the T cells that reside in this inguinal lymph node, in the skin draining lymph node. So that was nice. And now we try to take this system in a disease paradigm. And actually, we looked at it in many disease paradigms, but I'm starting with the most common one that a lot of people use to mimic inflammation, autoimmune inflammation in the brain, which is MOC peptide-induced EAE. So we have, again, our T cell conditional mitodendra. This is this photoconvertible protein expressing mice. And we just induced EAE in them by subcutaneous, in this case, immunization. And I'll come back to this, what the implication of this is. And then after some time, in this case, after 2 days, you can, but we did a lot of time kinetics. You can actually do it at later time points, not too late, because otherwise you will lose the label. We came back and analyzed where our activated cells, or our labeled cells in this activated paradigm would reside. And you can see here, when you, in this case, irradiate the skin draining lymph node, the inguinal lymph node, you can still see quite a bit of these activated, or quite a few of these activated T cells in the inguinal lymph node, but basically nothing, or very minor, only very few dots in the alternative lymph node site, suggesting that the activated cells in the inguinal lymph node would not traffic to the alternative lymph node, but would probably then go to remote places. And I'll come back to that later. Now you can do the experiment the other way around. So you do the same immunization. You label, in this case now, the mesenteric lymph node. And again, there is no cross-trafficking. And that is good, because that would mean that otherwise your system would be very leaky, and it would be hard to say where, when you pick your labeled cells, whether they would not have gone to the alternative lymph node site. But that does not appear to be the case, at least not to a great extent. So now the big question is, when we now label these cells in that manner, can we find them back in remote places, so in remote tissues, like in the spleen, or in the central nervous system even? And I can tell you, we found it in the spleen, and we also found them in the central nervous system. So this is clearly a fraction of photoconverted T cells here, CD4 T cells, that we find in the brain, in the central nervous system, after inguinal irradiation. And we call them IT cells because they were labeled in the inguinal lymph nodes. We can do the same with labeling in the mesenteric lymph node. And again, we find T cells that are labeled in the brain that most likely now we know for sure that they come from the mesenteric lymph node. We call them M T cells. And of course, so that particular disease paradigm is based on subcutaneous immunization. So it's likely that the skin draining lymph nodes are the draining lymph nodes in a way. But when we looked at the antigen specificity of these labeled T cells in the CNS, we found, as you can see here, that's no surprise that if they come from the inguinal lymph nodes, they bind the MOC tetramer, not the control tetramer, but they also do this when they come from the mesenteric lymph node. And that's probably to do that by this subcutaneous immunization introduces massive amounts of antigen that kind of spills over also to mesenteric lymph nodes. So you would have some priming, and we did a lot of experiments to this and can actually show this also in the mesenteric lymph nodes. So you would have MOC-specific priming in the mesenteric lymph nodes as well. So with this now, with this setup of the experimental system, we can start asking our real questions. So, and this is the way how we did this. So we had basically two groups of mice, one with inguinal T cells labeled and one with mesenteric T cells labeled. And then we picked only the labeled T cells in remote tissues, in particular from the spleen and the central nervous system. And we hashtagged them so we could analyze them together in order to avoid batch effects. And then we could computationally resort them again based and look at their single cell transcriptomes. This is what we did first. And since we physically labeled them, we could exactly know whether these are I T cells coming from inguinal skin draining lymph nodes or M T cells coming from the mesenteric lymph nodes. Okay, so first thing we did, we looked at the T cell receptor. So because that is, if you want, an endogenous barcode that those T cells have. And here the first finding was, it's very clear that identical clones are only found within each stream. So we find clones in the eye stream in all organs, so in the skin draining lymph nodes in the spleen and in the CNS, and we find private clones also in the M stream, so in the mesenteric lymph nodes in the spleen and in the central nervous system, and we do not find public clones. So we never found basically a clone that was actually that popped up in the two streams, in the I stream and in the M stream. However, what we did find was that some T cell receptor V-beta families, in this case, were actually shared between streams. So if you look here, down here, for example, the V-beta 5 or V-beta 12 or 19 or 20, they were actually shared and also expanded in both streams. And another one was, in this case, V-beta 3, which was also shared between I and M. So suggesting that there is common families of V-beta receptors. And since there is a big literature on the sequences of the T cell receptor that would recognize MOC, we dug a bit into this and found that this is actually most likely associated with MOC reactivity in this case, suggesting that there is MOC reactive cells that we had already seen with the tetramer staining in both streams, but there is no public clones. There's only private clones within each stream. But now when we look at the transcriptomes or at the phenotypic properties of these cells, so what can we find? So maybe let's focus here on this cluster, which is basically the cluster of T cells that we picked from the central nervous system. And you can already imagine or can see here that there is a bit of overlap, but there's also distinct features, so distinct clusters in this CNS residing T cells based on whether the T cells come from the skin or the gut draining lymph nodes. And when we now put onto this known signatures of T helper cells, you can see that maybe the iT cells are closer related to Th1-like cells. That's maybe something you could say in the central nervous system. But eventually, it's a bit all over the place, in particular for Th2, but also for Th17 and also for regulatory T cells. So it doesn't really, you can't say that Th1 cells are primed in skin draining or Th17 cells are primed in mesenteric lymph nodes, but it's a bit of a mixed picture here, as we had already imagined and as Christoph and Diane had already seen in their infection paradigm. So the question was, can we now, based on the provenance of these T cells, iT cells or MT cells, define signatures that would define these cells? And this is what we tried. So we tried to look at the genes that were always upregulated in iT cells versus MT cells in all the tissues. And we came up with these core signature genes here, about 100 for iT cells, about 50 for MT cells. This is just a few examples. So this chemokine receptor 6 is always higher in iT cells, while this purinergic receptor P2RX7 is always higher in MT cells. And here some integrins, integrin alpha 4 always higher in MT cells, and integrin beta 1 always higher in iT cells. And there's a lot of many genes that are very interesting that pop up in these signatures. So a bit of a follow-up question is, now how is this regulated? And we did ATEC sequencing also on these cells, and we found that the open chromatin areas are also distinct between iT cells and MT cells. And what you can do, and this is something we still have to do, and we are following up on this, is now which, that would be our ultimate goal, which transcription factors or transcriptional modules are responsible then for these distinct signatures. So asking the question, which transcription factors bind on these differentially open chromatin areas? And you can see here in this waterfall plot that appears to be quite distinct again for MT cells here in orange and iT cells here in purple. So you have, for example, this one, TCF7, which appears to be somewhat more active in MT cells, while, for example, TBET or RANKS1, even though this is not clearly at the edge of the spectrum, appear to be more active in iT cells, so in skin primed T cells. This is certainly something we still have to work on and really come up with maybe a hierarchy of transcription factors that define one or the other phenotype of these cells. So of course, a concern that you can have is, so this is a system which is very much based on subcutaneous immunization. So is this a universal feature, or is this really something which is very much driven by the subcutaneous route of antigen administration, or even the adjuvant that we use? So we did the same thing here with an IV priming system, where the antigen is certainly not biased in terms of mesenteric lymph nodes versus skin draining lymph nodes. And we basically find the same markers to pop up. So we have, this is just a few examples here. CXCR6 is actually higher in iT cells, so in lymph nodes that drain the skin, even if we give the antigen IV. This purinergic receptor is higher expressed in T cells that have been labeled in mesenteric lymph nodes. And this is just the two integrins that I talked about, which are also kind of distinctive in iT cells versus MT cells in this IV priming system. And the nice part is we even then used a system which doesn't rely on immunization at all, which is the spontaneous EAE model that Hartmut Weckele and Vijay also introduced, which is basically a double transgenic mouse that has a MOC-specific T cell repertoire and a MOC-specific B cell repertoire. So you don't have to immunize these mice, so they get spontaneous EAE. And we crossed our, photoconvertible system into this background, which was quite a bit of crossing, but then did the same experiment. So we labeled the cells either in skin lymph nodes or in mesenteric lymph nodes, and then in this case, picked them from the spleen. And you can see that cum granosalis, it's the same pattern, looking at some of these markers, but also globally. When we did RNA sequencing, we could re-find the markers that we had defined as I T cell markers versus M T cell markers, also in this spontaneous EAE model, which suggests that it's kind of likely that the properties, the milieu of these different anatomical sites of priming in either the gut or the skin draining lymph nodes define or dictate the properties of these effector cells that we then later find in the target tissue, in the spleen, or in the target tissue in the central nervous system. So the big question now is, so is this associated with biologically meaningful differences in the effector functions of these cells? And the only way to address this is to do an adoptive transfer model with either I T cells or M T cells. And this is what we tried here. So we induced I T cells, and the way we did this is we actually immunized photoconvertible mice, labeled the inguinal lymph nodes, and then sorted, in this case, the labeled lymph nodes from the spleen. So we could make sure that these are for sure only I T cells, and transferred them into secondary hosts. We did the same thing for M T cells, and then looked at what happened to the secondary hosts. So we don't have mixed priming in the secondary host, but we only transferred either I or M T cells. And what was kind of obvious already by just looking at this was that the cells that had been primed in skin draining lymph nodes, I T cells, were kind of able to infiltrate both the gray and the white matter. So that is, when a mouse gets severely sick, it's always the case that you find also T cell infiltrates in the gray matter here in the spinal cord, not only in the white matter. But when we transferred M T cells, we didn't see this pattern. We always had the feeling, and we could actually also measure this, of course, that these infiltrates were very much restricted to the white matter. So in this case, in the spinal cord, white matter is of course at the outside of the tissue here in the white matter tracts. This is just a blow up of this, suggesting, and that was actually quite, in my mind, an interesting finding, that this homing pattern of I T cells and M T cells in the central nervous tissue appeared to be different. And one determinant of this, even though I think it's not the only one, could actually be the expression of this chemokine receptor CXCR6, which is higher expressed in I T cells than in M T cells. And because that had been reported, even though I think not published, but I think I heard it in an abstract at some point, that CXCR6 knockout mice, they do get sick, they do get EAE, but when we looked at them histologically, the infiltrates were very much also again restricted to the white matter, while in the wild type situation, that's been known before, you have white and gray matter infiltration in these mice, suggesting that really CXCR6 could be one of the determinants of this. So, okay, coming back to the starting point of this is this something we can exploit for patients? And we try to start to understand this by looking at CD4 T cells of multiple sclerosis patients that we isolated from the spinal fluid, from the cerebral spinal fluid, because this was very close to the central nervous system. And we basically checked whether we could re-find our I signatures versus M signatures also in these CD4 T cells of MS patients. And so we did single cell RNA sequencing of these T cells, and we just focus on CD4 T cells here. And this is the UMAP, I know the Tisney plot of this experiment. And you can, okay, so you can see there's probably 4 clusters here that, yeah, overlaps not entirely well with expanded versus non-expanded T cells in these CD4 cells of the CSF, and very clearly does not at all really coincide with known T helper cell signatures. If you overlay them over the single cell maps, you can see that's all over the place, in particular for Th17, maybe a bit less for Tregs. They are more specific to cluster 3 here, but not really for Th1 and Th17. However, when you now look at our signatures that we defined actually in the mouse system, I T cell signatures versus M T cell signatures, that appears to be, even though we still have to do more tests, more consistent with the clusters that we find in these single cell RNA-seq analyses of patients. So this one I introduced to you already, 606, is very much focused on this cluster 2, and this one, CD73, is very much focused on this cluster 0, for example, and that would be part of the M T cell signature, and that is part of the I T cell signature. So the idea would be that by defining these signatures, we can then maybe stratify also our patients in terms of where, for example, whether they have more gray matter versus white matter lesions. I mean, that would be the ideal situation, but we are still not there. So to summarize this, and I have some discussion slides now then at the end. So I will just quickly go through this because I told you all this. So MOC-specific T cells are primed in skin draining and gut draining lymph nodes. So we call them I and M T cells in this case. We have seen that they produce private MOC-specific clones, no public ones, private ones. The transcriptomes of skin and gut-primed encephalidogenic T cells are distinct, but cannot really assign to known T helper cell subsets, basically. We are now trying to identify better, this is a process which is ongoing, the transcriptional modules that may dictate these signatures, I T cell versus M T cell signature. And we think that this is very much also correlated with distinct effector functions and homing patterns of these T cells in the central nervous system. And there might be some determinants that we may have identified already, like CXCR6, for example. So that is, to close the loop in a way, now a also targetable immune system intrinsic determinant of CNS pathology, we think. And now the question, of course, is where do we go from there? I mean, and this is something we are really very much thinking about every day. So how can we exploit this? Yeah, and so one big question that we have is, when we have priming of I T cells or M T cells, how is this, or what impact does this have on their fates in the target tissue or in remote tissues? So for example, in terms of their effector potential or their memory potential, or even their potential to become, for example, T follicular helper cells. And so based on the markers that we found in this experimental setup that I described to you, it's kind of likely that the capacity of these T cells to become either this or this is very different. And this is something I think we really need to understand better in order to also understand better the immunopathology that happens in the central nervous system. So this is just one last example, one last data slide that I want to share with you now. So we have these signatures, and I just put here kind of randomly 6 markers that we identified here, so that are distinct between I T cells and M T cells. So this is, for example, IL-18 receptor, CXCR6, folate receptor 4, and so on. So a list of receptors. And when we use them and combine them, we can actually do pretty good I gating and a pretty good M gating. Yeah, and this is just now validated by using our photoconvertible mice. So when we, for example, do this kind of I gating just based on these surface markers, we can basically exclude M T cells. So we have only I T cells, and we can do the same thing when we do an M gating. If you want, we can basically exclude I T cells. So we have only M T cells. And when we now apply this surface, and that's a very easy experiment actually, and use, for example, reporter mice for, say, FOXP3, you can actually see that in the spleen, at least, that's where we did the experiment, but it's kind of likely that it's also the same in the central nervous system. This segregates very well with the fraction of FOXP3 expressing T cells. So in the M-gated cells, yeah, they have a good fraction of FOXP3 positive T cells here based on the FOXP3 reporter, and there's almost nothing in the IT cells. And that is something that we like a lot, because that's a very interesting, because that's a way to really look at the fates of these T cells that have been primed in different sites. And I think that, at least in our mind, has quite a bit of a potential to look at and to exploit it for fates of these T cells in remote tissues. So this is the very last slide. This is our concept. So we think that the site of priming determines very much later functions of these T cells. And we think that we actually find, we showed in my mind, that we find I T cells and M T cells in the target tissue. And so Vijay actually, I mean, he also obviously has been interested in this, even this was really totally independent. And you may have seen that he came up with a paper recently where he has a very similar concept. But he thinks that these differentially primed T cells that are, maybe MOC specific, are kind of converted to a more pathogenic species in the spleen. He thinks it's because they sense IL-23 there, and only those pathogenic ones would then go to the target tissue and induce immunopathology. I think this is where we differ a bit. I think that our data really show that we also find M T cells in the brain, and we don't think that they are totally harmless. But I think the jury is out, and we will have to see whether this kind of tissue imprinting holds up. I think, in my mind, it does, and can, as I started out with, be exploited for stratifying patients and also coming up maybe with very specific T-cell based therapies for patients with multiple sclerosis. But of course, this is universal, that can basically also be applied to other autoimmune diseases that happen in remote tissues. So with this, I really want to thank you for your attention, and also, of course, the people that did the work. And I just want to highlight very much Michael Hiltensberger, who was spearheaded the project in my lab and had help, particularly with the single cell analysis from Eduardo Beltran from the LMU Munich. These are our local collaborators and our funding sources, and I really thank you very much, and I'm happy to answer the questions in the Twitter account. Thanks.
Thank you very, very much, Thomas. This was an amazing talk, very cerebral, wonderfully put together, super exciting. I did like, especially in the end, your discussion points. I am certain there will be a lot of people reaching out to you and will ask questions and want to discuss more with you. The way to do it, I hope I'm sharing my screen right now, is to go on Twitter. You go look for Global IMMUNOTALKS, and then you can ask questions for Thomas Korn, and he will answer under ImmunoSpeaker, and that's how you'll find him. And I'm sure this will be a lively discussion. Very excited, and thank you very, very much, Thomas, for this wonderful presentation, and everyone else for joining. Thank you, and good evening, good morning, and good afternoon. Bye.
Thanks for having me. Thank you.